Hi everybody. In this video, we are almost ready to start talking about the mechanism of DNA replication. So, mechanism of DNA replication. And I like to talk about this mechanism by describing 6 steps. But before we do that, I think it might be best to first look at the properties of 2 DNA polymerases in E. coli. We are going to talk about DNA replication in E. coli. It works very similarly in eukaryotes and prokaryotes, and it's just easier to describe the process in E. coli. So we are going to talk about 2 different enzymes called DNA polymerases that are DNA polymerases. And polymerase, to polymerize something, is to take smaller units of something and join them together and to make a polymer. So if these are nucleotides, a polymerase would join them together. It doesn't quite work like that, but we're essentially joining, DNA polymerases join DNA nucleotides together. So these 2 polymerases have a couple of properties. So one of the properties they do not have is called initiation of synthesis or initiation of polymerization. So they cannot just take a random bunch of nucleotides and start joining them together. As we will see, they need a free 3' end of another nucleotide, or a primer, I should say. And we'll talk more about that in a moment. There is also an activity called a 5' to 3' polymerization activity. So 5' to 3' synthesis. Sometimes I call it polymerization, sometimes I call it synthesis. It means the same thing. So they both have that property. There is a property called 5' to 3' exonuclease activity. Now that means if the enzyme encounters another nucleotide, say while it's using a template to synthesize DNA, if there is a nucleotide in its way, it can cut out that nucleotide and keep synthesizing DNA. So DNA polymerase 1 has that process. DNA polymerase 3 does not have that property. There's also a 3' to 5' exonuclease activity. And both of the enzymes have this activity. And this activity, as we will see in, I think, a future video, is involved in proofreading. So if a polymerase makes a mistake, adding the wrong nucleotide, this property allows it to cut out that nucleotide and fix its mistake. So for this video, what I want to focus on is these 2 things right here. So what does 5' to 3' synthesis mean? How come these polymerases can't just initiate synthesis on their own? And to see why that, to understand those things, what we need to do is review the structure of a nucleotide. So in a previous video, we talked about the structure of a nucleotide, which has a nitrogenous base, a 5-carbon sugar, and a phosphate group. So let me diagram the 5-carbon sugar. And remember, this is the 1' carbon, this is 2', 3', 4', and then 5' will be up here. And I'll put the C in here, so 5'. So these angles right here represent carbons. This is an oxygen. This 1' carbon, this is the one that binds the nitrogenous, is bound to the nitrogenous base. And this one, this carbon, 5' carbon, is bound to a phosphate group. And I'm going to put a phosphate group here. So phosphate with 4 oxygens. Now this 3' carbon here is bound to an OH group. And I'm going to diagram it like that. Now, what DNA polymerase does when it's joining nucleotides. Now, I can diagram a template in here. So let's say there's a template here, and this base is bound to that base here. And I won't diagram the nucleotide over here. And there's a base over here, and I'm not diagramming the nucleotide. Now what the polymerase is going to do is a nucleotide is going to come in, and if it's the right nucleotide, it will hydrogen bond with this base right here. But it's not just any old nucleotide. So the nucleotide that's coming in here has to be what we call a DNTP. So D for deoxyribonucleotide, because this is DNA. NTP is nucleotide triphosphate. So what that means is that the nucleotide that is coming in here needs to have, now how can I diagram this? I think I need an eraser here. Needs to have 3 phosphates, or phosphate groups. And this is how the phosphate groups look when they're connected to one another. So they have single oxygens connecting them. And then this is connected to the 5' carbon of the nucleotide. And this has your free OH group attached to the 3' end. And then this is the guy here that's attached to the base. So for this diagram, let's just ignore this for now. Just ignore that. I just want to point out how this 3' OH group, or this 3', this oxygen bound to this 3' end, is going to be linked up or bound to this nucleotide by the polymerase. So essentially what the polymerase is going to do, it's going to orient these atoms in such a way that this oxygen right here, this guy's going to be lost. Let's just get rid of this guy. That guy's lost. But this oxygen right here is going to come in here and attack this phosphate right here. This thing's going to be lost. This bond is going to be broken. And as a result, this thing right here, which we call, why am I blanking on the name? Pyrophosphate, or PPI. This pyrophosphate is going to be lost. Oh, why am I'm going to have to look that up, make sure that is correct. But that is going to be lost. So, and this thing right here is going to be connected to that phosphate right there. So we have oxygen, phosphate group, CPOOO, so that's our phosphate group, attached to the 5' carbon. Now that's the phosphodiester linkage. So let me diagram this after this is joined. So I'll neaten it up for you and you can see what the result is. So here's the 3' end of the nucleotide that the polymerase is joining the new nucleotide to. Here's the base. It doesn't matter what it is because we are just curious as to how nucleotides are joined to this 3' end here. So like I said, remember this guy is lost. Oh, sorry. P-O-O-O. This is lost or recycled. Now this oxygen is now bound to the phosphate, and this is bound to the 5' carbon of the next nucleotide, the nucleotide that was just joined to this one by the polymerase. Okay, and now this has a free 3' OH group that can now be linked up or bound to the next nucleotide in the chain. So you can see we're moving from the 5' carbon right here, 5' end, in this direction. So we keep adding to the 3' end. So this is one reason why we can call the direction of synthesis as occurring in the 5' to 3' direction. Okay, now this bond right here, 3' carbon to oxygen to phosphate to oxygen to carbon, the 5' carbon, this is the 3' to 5' Phosphodiester linkage that is formed between adjacent nucleotides by DNA polymerase during the synthesis process. Okay, so the 3' to 5' phosphodiester linkage. So this whole process, this joining of this nucleotide to this nucleotide is performed by DNA polymerase. And we are going to see how this mechanism fits into the whole process of DNA synthesis in the next video.